Okay, thank you to the chairman for the introduction, and also I'd like to thank my friend Dimitrio for inviting me to this great meeting. So today my topic is about the therapeutic efficacy of anti-CD19 CAR T cell in a mouse model of SLE. So first of all, let me introduce the background of the disease. So as we know, the systematic lupus erythematosus, SLE, is an autoimmune disease which mostly affects the woman with the childbearing age. So as we know that the symptom of SLE is mostly like the rash on the skin, and also the proteinuria, and also some of the patient will suffer from the pain in the joint, and also some central nervous system syndromes. And as we know that the symptom of the disease is very diverse, and so we want to know how to treat them with a strategy that will be efficient to all the patients. So we know that the pathogenesis of the SLE is mostly like this. We know that our immune system will present the foreign antigens when the foreign antigen is exposed to our immune system. But when the body have some genetic susceptibility, and also with some danger triggers in the environment, such as the overdose UV or the virus infection, some autoantigens will exposed in our circulation. So in this way, that the T cell and B cell will be activated by our autoantigen. So then the B cell will generate a large amount of all the antibodies. So the autoantibodies will bind to our autoantigens, and together with the completant, so we will find a lot of immune complexes formed in the circulation. And along with the circulation, the immune complexes will deposit in the like the skins or the kidney. So, and then accumulatively, it will cause the dysfunction of the certain organs. So current treatment for the SLE is mostly based on the B cell targeting. We know that there are a lot of antibodies found to be functional in the treatment of the SLE. Mostly of them are B cell targeting. So, but also there are some small molecular inhibitors. But currently there is no format of cell-based treatment for the SLE. We want to try that whether with another form of the B cell targeting strategy will be better or be more efficient to all the SLE treatment. So we think about maybe we can use a CAR T cell to treat the SLE. As we know that the CAR T cell is human-made molecular, which is transferred to the T cells. And this molecular is like this, and it will have the scFv motif on the outside of the cell, and it will, when it binds to the target, it will have the signal 1 and signal 2 of the T cells. Mostly the signal 1 will be the CD3 signal, and the signal 2 will be CD28 or the 4-1BB signals. So, and then as we know that CD19 is one of the most frequently used target of the CAR T in the leukemia and the lymphoma treatment. So, and we also find that CD19 is one of the most widely expressed surface marker of B cells along in from the pre-B stage to the some of the plasma cells. So we want to try whether the anti-CD19 CAR T cell can treat the SLE. So first of all, we have generated a different type of the CAR construct. As we can see here, we have generated the CAR T cells with the second signal with 4-1BB or CD28, and we have also tried that with the first signal of CD3 with either the WT type or the first one and the third one the third ITAM motif inactivated form. So this is the certain types of CAR T cells found to be very useful in the cancer treatment. So first of all, we want to try whether this kind of CAR T cell will kill the B cells very efficiently. So we tried that with our constructed CARs. The CAR T cells can kill the B cell very efficiently with a very low ET ratio. So as we can see here, that the CAR with the construct of the first signal of mutated CD3 and the CD28 as the second signal have the best killing effect in the in vitro assay, indicated by both the specific lysis of the B cell and also the intracellular expression of interferon gamma. And so we use this kind of CAR T cells to test whether it can treat the mouse SLE. So as a mouse model, we use the MIL LPR mouse. This is a kind of mouse with a certain genetic background, and also the mouse carries a fast mutation, a 1-point mutation on the fast gene. So this mouse will have a large amount of B cell and T cells, because the T cell cannot be killed during the T cell development. So with the accumulation of T cell and B cells, the mouse will have the symptoms very similar to the human SLE syndromes. So with this kind of mouse, we generated the CAR T cells, and then we tried to use the therapeutic strategy very similar to the cancer treatment with low dose total body irradiation as a preconditioning. And also we use these CAR T cells at the age of the 13 weeks old. This is the kind of age that the mouse has already developed very severe symptoms. So as a controls, we use a mouse without the TBI, the low dose total body irradiation, and also we use a control as with the antibody injection. And then we will test the result at the age of 22 weeks, which half of the mice will die automatically with the very severe syndrome. So first of all, we test whether the B cell exists when the mouse received the CAR T cells. So from the flow cytometry data, we can find out that the free flow B cell almost diminished after 1 week of CAR T cell injection. So we can see here that the low rate of B cell persists until 9 weeks later. And also we found that the percentage of the plasma cell, which is indicated by the CD138, and we can see that from the start there is no change, but when the time goes by, the plasma cell generation is also goes lower in this treated mice. So also we tested whether the CAR T cell will function in long term in this kind of mouse. So we tested after 9 weeks of the CAR T injection, we used in vivo and in vitro test to see whether the capacity of killing B cell remained in the spleen of this mouse. We found that a certain killing capacity still remains, but the T cell cannot be found because maybe the mouse already have a lot of dysfunctional T cells in the free flow. So then we also tested whether the symptoms of the SLE has been altered after the treatment of the CAR T cells. So we can see that from the total survival rate, the red line indicates the treatment group. We can see that all the mice survived until 22 weeks, and half of the mice has died at the week of 22. at the age of 22 weeks. And also we can see from the clinical scores and the size of the spleen, we can see that the treated group showed a great, showed better condition than the control groups. And also we have tested we have tested whether the function of the kidney is still good in this stage. We can see that from the slides, after the treatment of the CAR T cells, the glomerulus seems to be much better than the control groups. As we can see here, there is a lot of accumulation of the lymphocytes, and also the structure of the glomerulus is changed here, but the treated groups remains better. And also we can see from the IHC slides to test whether the immune complex will deposit in the kidney. We can see that the CAR T treated group showed much better and showed less immune complex deposition.